Okay, let's quickly look at the antibodies of the adaptive immune system, the humoral immunity. Right, so the structure of the antibodies, we'll be using one kind of antibodies as the representative of a basic structure, right? So this kind of antibody is known as IgG, right? Later when we look at different kinds of antibodies, that would make more sense. Okay, but a typical antibody molecule, right, would have such a structure, right? It is a quaternary protein that is made up of 4 polypeptides, 2 light chains, as you can see here, and 2 heavy chains. If I just use my laser here, 2 light chains, so there are shorter ones, and 2 heavy chains, which are the longer ones. All right, the light chains are identical, and the heavy chains are identical as well, right? So the light chains would consist of 2 domains. Domains are just like portions or parts of one polypeptide. So we call them domains, right? So we have this variable domain and a constant domain in each of the light chain polypeptide. And also for the heavy chain polypeptide, there's also a variable domain as well as 3 to 4 constant domains. Okay, and the light chains and heavy chains are joined to each other via disulfide bonds. Okay, as you can see with the SS here. All right. On each basic structure of the antibodies, there are 2 identical antigen binding sites. Identical because, again, right, if you look at the antigen binding site, it is formed by the variable regions of 1 light chain and 1 heavy chain. Right, so again, each antigen binding site is formed by the variable region of the heavy chain and the light chain. And the 2 heavy chains are identical. The 2 light chains are identical. So therefore, the 2 antigen binding sites are identical. Okay, so the antigen binding sites would be the binding sites for the antigen, obviously, right? And as we have looked at in the previous video, right, each of the antibodies produced by a different B cell will have a different specificity, right? So different antibodies will have a slightly different antigen binding site, okay, for a different epitope, okay. Right, so so here to show you the formation of the antigen binding site, right, with the variable region of the heavy chain and the variable region of the light chain. So it has a particular conformation, particular shape that is specific to a particular epitope, right, on different antigens. Okay. So on the antibody as well, right, in the heavy chain here at the end of the antibody, right, it is known as a constant region or a FC region, right, and it will bind to certain immune cells. Right, so the antigen binding site binds to antigen, right, on the pathogen. The constant or FC region would bind to immune cells that have these FC receptors. Okay. Right, so that is a basic structure of the antibodies. Next, we will look at the different types or different classes or isotypes of antibodies. Right, so again, right, just now we mentioned the structure that we looked at just now is following the structure of IgG, which is one of the most abundant kind of antibodies in our body. All right, but we have 4 others classes or types of antibodies. We have the IgM. This is the IgM, which is quite a big antibody, right? You can see there are basically a combination of different subunits of this basic structure. Okay, pretty cool, right? Then we have the IgA here is basically like a dimer, right, of this basic structure. Then the IgD and the IgE are somewhat more similar to the IgG. in terms of structure, but they have different functions, and they are produced by plasma cells at different times and different maturation level. Okay, as we go on in the topic, where you will learn all this a bit more. Yeah. So anyway, for the different classes of antibodies, they differ in their constant region, as you can see here, represented by all these different shapes of the constant region. Right, so constant region is here. Yes. Okay. So because the constant region is different, then they would be able to bind to different immune cells, because they will have different FC receptors. Like for example, the IgG, right, can bind to FC receptors on macrophages, and IgE can bind to FC receptors on eosinophils. Right, so they do recognize different immune cells. All right. So next up, for the list of function of antibodies, they basically bind to pathogens, right, or bind to antigens on pathogens via the antigen binding sites. And because of that, right, that brings about different results. Okay, so we can have neutralization to block the pathogens from infecting our cells. Right, so imagine, say, the influenza virus, for example, and you have an antibody that binds to the hemagglutinin. Right, so therefore, the hemagglutinin cannot bind to our specific receptors, right, or with salicylic acid on the epithelial cells of the respiratory tract. So therefore, there's no infection then, if we have the successful binding of the antibodies. Right, so this is known as neutralization. Then next we have the opsonization. That's when the antibodies bind to pathogens, and then especially for IgG, right, they also bind to the macrophages, right, via their constant region, right. So that facilitates the macrophages phagocytosing. Yeah, this is the term, right? Phagocytosing the pathogen. Right, so this phagocytosis is aided by the antibody. So this is known as opsonization. Okay, then we have agglutination, right? Because each antibody at least have 2 antigen binding site, right? And for the IgM, for example, it has like much more. So each antibody at least can bind to 2 epitopes on 2 different antigens, maybe to 2 different pathogens, right? So that brings the pathogens together, right, for more efficient clearance. And for soluble antigens like toxins from bacteria, for example, right, the binding of antibodies to the soluble antigens can make it insoluble, and therefore make the antigen, or maybe toxin, right, lose its activity. And then, right, the antibodies can also induce the release of histamines by the mast cells or the basophils, and that would then enhance inflammation. All right. So again, this is via the binding of the constant region to the FC receptors on the basophils and mast cells. And one last one, right? The antibodies can also activate the complement system, right? So that would make the clearance and killing of the pathogens more efficient. Okay. But our main focus really would be on the neutralization, opsonization, and agglutination. These are the main focus that we will have for the functions of antibodies. But the rest are also legit functions, right? You can cite them as examples as well. But in the tutorial, for example, our focus will be more on the neutralization, opsonization, and agglutination. Okay. All right, so anyway, these are the functions, the antibodies structure as well.